Merci, Monsieur le Président de séance, et merci aux organisateurs pour cette journée et pour l'occasion qui m'est donnée de pouvoir présenter nos travaux sur cette fameuse épigénétique transgénérationnelle. So before I start, maybe just to remind everyone that indeed DNA does produce RNA, does produce proteins, so there is nothing without DNA. We don't have epigenetics without DNA. That's basically a first point. And then when we go to DNA methylation, which will come as a recurrent theme, one needs to remember that DNA methylation is not universal. It is found throughout the eukaryotic and prokaryotic kingdoms, but you can find whole taxa which are devoid of DNA methylation. So you can live happily without DNA methylation as well. Having said that, of course, DNA methylation is very important, at least in two groups of organisms, plants and mammals. And in plants, now I need to go to observations, which are actually, for the first one, very old, initially made by Linnaeus 250 years ago. Of course, he was not talking about mutation at that time. There was no such thing as genetics, as alleles, mutants, loci. There was just variation, and he was cataloging, species on Earth that the Creator, in his whole generosity, put on Earth, and he was very stuck because he found a plant which he called peloric, meaning monstrous in Greek, which looked very much like what he had described as Linaria vulgaris. But peloric has this very, very weird flower phenotype, with altered symmetry compared to the wild type, which is bilateral symmetry, and now you have this radial symmetry. It just turned out that we understand much more now about peloric in molecular terms, and when we had a way of going back to the gene, which probably was indeed affected or mutated, as we say now, in peloric compared to the wild type, we found no DNA sequence change. There is still DNA, but there is no DNA sequence change. What you have here is in peloric, you have a piece of DNA which is now hypermethylated and associated with silencing of the gene. And this hypermethylation, we don't know how it came about, whether it preceded or caused the silencing of the gene, but what is remarkable is that this was then transmitted, and is transmitted according to Mendel's laws. So again, that's another important point. You don't always have to oppose epigenetics and genetics. I remind you that genetics was invented long before we discovered that DNA is the repository of the hereditary material. So you can have genetics without change in DNA sequence. You always have DNA for sure, but you don't have to invoke a change in DNA sequence to get this mutant phenotype. So this is what now we call an epimutation. And I put on the right the structure of the nucleosome just to remind everyone that we're indeed dealing with just one mark here, DNA methylation, but which comes about with a whole bunch of other modifications which profoundly alter chromatin and the way it is read, expressed, permitting or not a gene to be expressed. Now you have below a much more contemporary example, which is called CNR. This is in tomato now. This is a non-ripening tomato compared to a wild-type tomato. And again, this is a very simple mutation, described in Mendelian terms as a recessive single locus mutation. So you could actually go to the locus and then sequence the mutant allele and the wild-type allele and try to compare the sequence. And you find the same DNA sequence. And yet you have genetics here. Genetics which is based, again, on hypermethylation in the case of the mutant, hypermethylation associated with silencing. What is important in this second example is that we know much more about the locus itself. We know that both in the wild type and in the mutant, we are now dealing with the same allele. That's genetics. I'm sorry to use these technical terms, but this is important. We are doing genetics here. So, oops, I'm sorry, what did I do? So we have the same allele, but we have two different epi-allelic forms. One which is epi-mutated compared to the wild-type epi-allele. Now what you see is that there is a repeat sequence upstream of this particular gene which is affected in CNR. And this repeat sequence, like most repeat sequences in mammals and in plants, are indeed the primary targets of DNA methylation. And so what happens is that in the wild-type, this sequence is methylated, but the promoter of the gene is not methylated. And now what you have in CNR is a kind of spreading of DNA methylation from the repeat sequence into the flanking gene sequence. And we believe that this may be causing the original epimutation. So we have perhaps here a mechanism by which you can create an epimutation. What we still don't know here is what triggered this spreading of DNA methylation from these sites in the repeat sequence into the flanking sequence, but at least we can propose the mechanisms by which we can create epimutations. Now, with everything that has to do with epigenetics across generations, there is the notion of stability, of course, because we know that epigenetics plays a very important role during development, and it is reset every generation. So what we have here is a failure to reset, an absence of resetting across generations. And yet, if you look carefully at both pyloric and CNR, you can see evidence of reversibility. In both cases, we are dealing with what we call somatic reversibility, namely that in the same plant, you will find flowers which are fully pyloric and flowers which are almost wild type. And now what is very interesting is that when you compare these different degrees of alterations, they correlate with different degrees of DNA methylation. With CNR, we have exactly the same story. Here we have a beautiful example of one cell very early on during plant development, during fruit development, that reverted from this fully methylated state to the unmethylated promoter state. And now all of the daughter cells of that particular cell, as the fruit grew, now, of course, have acquired this revertant state. And so now what you have is a somatic revertant sector at the surface of a fruit which is fully mutant otherwise. What is fascinating in this example, again, we know much more about CNRs than we know about pyloric, is that when you sow seeds collected from pyloric plants, you always reproduce pyloric. You never see any meiotic reversion, so you never revert that phenotype, and yet you readily see it at the surface of the fruit. So you have very unstable somatic transmission, so to speak, but extremely faithful meiotic transmission. It's the exact opposite of what you have in development, where you have very faithful mitotic transmission, and of course, very faithful reversion, reprogramming from generation to generation in the gametes or in the early embryo. So now, all of that to then ask questions, what is the potential for this kind of epimutations? And here, I'm within a framework of genetics. I'm not opposing genes to what, by the way? Genes are genes, okay? They were defined before DNA. So I'm opposing epigenetic variation to DNA sequence variation. I'm not opposing genetics to epigenetics. I'm asking, is there a dimension of epigenetics in genetics as we were taught from Mendel and so on and so forth? So what is the potential for epimutability genome-wide? And we believe, based on the example I gave you with CNR, that repeat sequences may be key to this particular aspect of the function of the genome. So, and we know that, of course, if you turn to the human genome, more than 50% of our genome is made up of repeats. So the potential for epimutability may be pretty high in organisms such as ours. Second, what determines the transgenerational stability of epigenetic variants? As I showed you with CNR, we have very faithful transmission across generations, and yet we can be unstable mitotically. And the third question is, of course, a very important one. How much of what we call natural variation, natural genetic variation, is indeed caused by something else than changes in DNA sequence? Could have a different explanation in terms of dynamic chromatin structure and so on and so forth. And finally, and this is a point which is extremely contentious to this day, is what role does the environment play in transgenerational epigenetics? And so we set out to try to understand, to address all of these questions using a model system, which is a plant again. But I have to also say one thing: this is not restricted to plants. It might be more complicated in mammals because there is very strong resetting of the mutation patterns in the germlines in mammals, unlike what we believe is happening in plants. Yet you can find examples in mammals as well. But this is an ideal system. First, we have a very small genome. And if you want to do epigenetics seriously, you have to resequence your genome. Because that's the only thing you want to oppose: sequence variation to chromatin variation. So unless you can know for sure that your genome sequence is the same as you started with, then you cannot invoke epigenetics. It's a possibility, but it's not a demonstration. Second, in this organism, we can perturb methylation patterns profoundly using mutants. And this, we are lucky, has no major consequences in terms of fertility or viability. So our plants are a bit altered, of course, yet we can now do genetics based on genomes which are drastically altered in their methylation patterns. And finally, Arabidopsis is a plant and grows actually quite widely across the world, and therefore we can then try to relate changes in epigenetic state that we may identify with differences in habitat, in climate conditions, etc., etc. Now, before I get into the heart of my talk, I need to say a few things about gene methylation in this organism, which is probably the organisms in which we understand best DNA methylation of all organisms, because indeed these genetics can be done because we have viable and fertile individuals. It's complex, and I will just highlight one thing, is that there is plenty of commonalities with what is known in mammals, but there are some differences. And one has to do with the presence of small RNAs, which by definition must be produced from DNA. These small RNAs, which are non-coding RNAs, play a very, very important role in directing DNA methylation to repeat sequences. And I remind you, repeat sequences are probably at the heart of the kind of epigenetic phenomena we are looking at here. So we used one particular factor in this complex machinery called DDM1, which is actually a very interesting factor. It's a nucleosome remodeler. So it basically moves around the nucleosomes. And by doing so, if you alter its function, you actually break down the methylation machinery altogether. So basically, you lose methylation of the repeat sequences. So we have used a mutant in a DDM1 gene to try to create a kind of genetics which is purely based on epigenetic variation, with no DNA sequence variation. And the scheme is here. I apologize, but this is very simple genetics. Mendel will have followed me very, very readily here. You start with a mutant, DDM1, in the same strain as your wild type. The strain is called Columbia. This is a strain that was actually initially sequenced in Arabidopsis, so we have a beautiful genome sequence to start with. And so the two parents basically have the same genome, except for the DD1 mutation, and perhaps, I don't have time to go there, but transposons start to be mobilized because you play with methylation, which is a key component of repressing transposons across the genome. And from this, you get your F1 progeny, and of course, now what you have is a genome which is by and large hypomethylated at repeat sequences compared to wild-type genome, and now you can recombine these two epigenomes, as we will call them, like you will do with two genomes that were different in terms of DNA sequence. And so we just backcross it to wild type in order to get rid of the DD1 mutation. We use the DD1 mutation only as an inducer of a transient change in DNA methylation. And then we ask the question, can this inducer, which is only there for a short time, can this inducer create changes that will have long-term consequences? So could we see the change in DNA methylation that were induced in this generation? Can we see it 8 or 9 generations further down the propagation? So what we did from this backcross progeny is self all our individuals until we can reach what is called homozygosity. But here, of course, homozygosity is only at the epigenetic level. Remember that all of these genomes are the same. So we have no sequence variation, except perhaps for a few transposons, but we have perhaps different epigenomes. And if now this falls into Mendelian genetics, segregation should follow Mendel's laws, and we expect, therefore, a very specific ratio of unmethylated to methylated alleles at a given locus in this population. So we did that, and that's a lot of work, as you can imagine, and we did that for hundreds of lines. And to summarize, what we had done initially was to characterize a contrasted set of regions for which we had fully methylated state in wild type and unmethylated in a DD1 mutant background. And we simply ask how many of these 3,000 DMRs, as we call them, differentially methylated regions, how many of these differences in epigenetic state that we created initially, how many of these differences are recovered 8 generations after we remove the inducer? So that's really the key question in epigenetics. You induce a chromatin change. Can it persist for long, independent of what was used to induce it? And to cut a very long story short, and I apologize, this is becoming a bit molecular here, what we found is that about a third of our DMRs, of our parental DMRs, were recovered in Mendelian proportions. So by themselves, they seem to be inherited according to what Mendel told us. They don't mix, they segregate in a 1-to-3 ratio, because that's what we were expecting based on our backcross scheme. And on the other hand, we have about 2,000 DMRs, which now tend to go back to their wild-type methylated state. And we know molecularly a lot about these differences, and it has to do with the targeting of these sequences with small RNAs. I don't have time to go into much more detail here, but suffice to say that now we have a very comprehensive understanding of the differential stabilities of epigenetic states that we induce across generation. Now, this is all done in the lab. And of course, the first question that we ask is this any relevant to what may happen in nature? And we were very fortunate 2 years ago to have at our disposal a collection of 130 strains of Arabidopsis taken from all across the globe, for which we had DNA methylation information at a single nucleotide resolution. So 130 accessions taken from all over the globe, for which we have now DNA methylation information. And what was found is, as we predicted, genes tend to be not methylated in Arabidopsis, but sometimes, and here it's a heat map, so what you have in red is genes or sequences which are not methylated, and in green, sequences which are methylated. Okay? So in most accessions, genes don't seem to be methylated, therefore you have this dense color, red color, but sometimes you see a gene which acquires methylation in one strain compared to the others. For transposons, of course, this is the opposite. We know that transposons are the primary targets of DNA methylation, so they do exist by and large in the methylated states, but sometimes you find one strain or two strains in which now this particular transposon has lots is methylation. So you have differential methylation also in nature. That's the first thing. And then when we do, what we did was simply to compare our DMRs, the one that we induced in the lab, with these regions of differential methylation in nature. And sure enough, what we found is that when we consider our stable DMRs, the one for which we have persistence across many generations, we have a very good overlap with natural DMRs. So we can say actually one thing now, is that these DMRs that are occurring in nature, together with a lot of DNA sequence variation, the two go hand in hand there. Well, we can propose that they may exist by themselves, because in the lab we have created them, independent of DNA sequence variation. And what is very interesting is that now if we take our reverting DMRs, the one that tend to regain DNA methylation very rapidly, you expect them to be less frequently found in the hypomethylated state in nature, because they have this inherent capacity to regain DNA methylation. And that's exactly what we found. This is why we have the least overlap with DMRs in nature. So all of that to say that our work is definitely leading towards DMR existing and being relevant in nature. And we can do genetics with them. And I'm going to skip that, I'm sorry, because I don't have time. We can actually derive genetic maps based on epigenetic variation. And there is nothing miracle or odd or weird about this. You know, we had DNA sequence variation, but here we have epigenetic variation. But of course, because we ask it to be behaving according to Mendel's laws, first we have them, then they follow all of the Mendelian rules. You can derive genetic maps, get crossing overs with this kind of variation, for sure. So the question that way, of course, that now it becomes very important for us, is to see whether we can relate that to heritable phenotypic variation. Right now it's only molecular. Can we relate that to phenotypic variation that is inherited in this population? So we did this experiment, and we were very careful to do our experiments in many different conditions, in a greenhouse, in a growth chamber, as well as in open fields, to see whether the epigenetic changes that we induced in the lab were stable enough to persist and to be robust against environmental perturbations or different environmental influences. And again, to cut a long story short, suffice to say that we find variation in all the traits we look at, and part of this variation we can measure has a genetic basis. When I say a genetic basis, doesn't mean that it's DNA variation or epigenetic variation. It has a genetic basis. It is transmitted across generations. That's the whole point we need to make here. And again, genetic quantitative genetics was built long before we knew that DNA is a support of the hereditary material. So then what we do is a very simple approach, which is called QTL mapping, quantitative loci mapping. We try to associate differences in phenotype, which we know are partly inherited, with differences in methylation states, which are by themselves inherited. And when we do that, we get our QTL with no problem. So here I'm just showing one example for flowering time. You have QTL on chromosome 1, on chromosome 4, and chromosome 5. The point of importance here is that, as I told you, we grew our plants in a greenhouse as well as in an open field. And we have the same QTL intervals, indicating that the epigenetic variation that we created in the lab is robust enough to exist also in an open field. It's not something evanescent that will disappear as soon as you start to change your environmental conditions. This is maybe not as hardwired as DNA sequence variation, but it is pretty solid and stable. Yet, if you look very carefully, and again, I don't have much time to go over this, we find even when we define stable DMR, so these DMRs that can be transmitted for at least 8 generations, we find evidence of sporadic reversion. And now the rates of sporadic reversions are much higher than what you get with DNA sequence mutations. So we believe that in the absence of selection, all of these genetic variations that we have created through epigenetic perturbation will progressively disappear over time. After maybe 100 or 1,000 generations, we will be back to square one, to the methylated state. But you can still do a lot of genetics in 1,000 generations. I remind you that there are not too many generations separating us from Christ or whoever, or even Neanderthal for that matter. So there is genetics, but perhaps with a finite time associated with it. And this translates into this scheme where our stable DMRs ultimately may get back to the methylated state. And that makes a lot of sense based on all kind of reasoning, which I have no time to describe today. Now, the last point, and the one that really hurts, is the environment. So we took a very painful approach to try to address this issue using extremely well-controlled conditions. We have this beautiful phenoscope, which was developed in Versailles, which is a conveyor belt, in a way, where all your plants, travel across the entire surface of your growth chamber. So they are all exactly under the same environmental conditions, except that some are watered and others are not watered. So we can have a water deficit or a drought stress applied to these plants. And I'm just going to summarize in one sentence. It failed lamentably. We haven't created epigenetic variation through this particular set of experiments, which is not to say that the environment will not induce epigenetic changes, but it probably requires much more drastic environmental challenges than what we applied when we did this experiment. So again, we are trying to explore this, but this is much more complicated now because we are going to deal with a lot of lethality to start with, and this is going to have an impact on the way we interpret our results as well. Now, one last point of caution. So as I told you, the environment is not a straightforward answer, for sure. One last point of caution. This is a beautiful work that one of my postdocs, Leandro Quadrana, produced some months ago now. We revisited these natural DMRs, the one that were described based on comparison of the methylomes of these 130 accessions taken from the wild. And what we discovered is actually very often when people talked about DMRs within genes, what we found is that it's not a DMR within a gene. Of course, you have methylation in this particular gene compared to all of the other accessions which have no methylation, and the same here. But what you have on top of it is a transposon that just landed there. And so what you have is exactly what I described for CNR. You have a transposon that acquires DNA methylation, and methylation spreads on both sides of the gene sequence. So what you have is a DNA sequence mutation. Whether you have epigenetic variation is another question. So far, we don't have evidence. It is methylated. I don't have the exact same allele in the unmethylated state. Of course, I can assume that it will happen just the same way as it happened with CNR. But again, this is a word of caution. When you see a difference in methylation, especially based on the type of analysis we do now with the short-read sequencing, you better wonder whether you're not dealing with DNA-based alterations, which are then translated into some differences in methylation states. So to conclude, well, I'm going to thank people first, if you don't mind. I want to thank, of course, a whole group of people because, as you can imagine, this first was a long endeavor, more than 10 years of work, and it involves people who are, of course, versed into phenotyping, high-throughput phenotyping, quantitative genetics, molecular biology, etc., etc. So the names are here. And I'm going to finish with these take-home messages, which you can read happily when I answer questions. Thank you very much.
Thank you. Questions, comments? Moshe.
Vincent, je devais connaître, mais je ne sais pas. Est-ce que dans les plantes, il y a une vague de déméthylation au cours de l'embryogenèse, comme chez la souris ou chez l'homme?
So I very briefly alluded to that. Clearly, we know that in mammals there is huge waves of demethylation and remethylation, both in the germlines and the early embryo, starting from the zygote. In plants, we have so far evidence of massive demethylation in one tissue called the endosperm, which is the nourishing tissue that surrounds the embryo within the seed. But this is a dead-end tissue, so we don't have reprogramming. We have a one-way demethylation. Now, on the paternal side, there is a few things happening, mostly towards at the level of repeat sequences again. And now we just actually produce some results which indicate that there is some dynamics in during embryogenesis. So basically, we see a kind of peak of methylation in the mature embryo compared to the early embryo or to what happens later. But again, it's a peak of methylation, but it's not extravagant, and we don't see a wave of demethylation or remethylation. So we just go perhaps higher than what we have in any other tissue, and this higher level is somehow transient, and we don't know what it does. Yeah. So there is little reprogramming in one word, in plants.
I just wanted to ask a question that was, in fact, double. What about the windows of exposure that we really see in mammals, and also the permanence of exposure over several generations?
Yeah.
Do you see?
So first, you realize that what we did initially was to use this mutation. So now every cell is exposed, including the germline, which comes late in plant development compared to mammals. So everything was exposed to DDM1, but transiently. We then remove the mutation. Now with our experimental design to try to probe the environmental influences, we were very careful. We wanted actually to do it in vegetative plants, so they have not yet flowered, to see whether they could remember something that is somatic, but the same cell that make leaves will then make the flowers. So the germline is actually contained within the vegetative part of the plant. And we saw nothing. So now we are asking the question, could we see something if we now apply the stress straight to flowering plants? And there was another question as well. I believe that we will not. But we have evidence that you could have cumulative effects from one generation to the next. That's called intergenerational phenotypic plasticity. We have that in plants for sure. Now the issue is to go beyond these intergenerational effects, which are just parental effects, put more simply, and see whether they can be transmitted for multiple generations. And there we are in a complete black.
Giacomo Cavalli.
It's very, very beautiful, Vincent. I had a question. In principle, you could envisage mechanisms that instead of using DDM1, use any other chromatin modifier gene that might then indirectly affect methylation. And I don't know to what point. I wanted to ask you, to what point have you explored that? Have you tried other mutants?
Well, we did. It took us 10 years to get to where we are. So I'm not going to do it again, if that's your question. But you're welcome to try it, and you will get results for sure. Now, I need to add one thing here. We have absolutely no clue whatsoever as to what created these DMRs in nature. Yet they overlap very nicely with our DDM1-induced DMRs for good reasons. They are transposon sequences. Some of them are fixed in the genome. So you expect them to be there. But what creates this variation in nature? Again, we are completely in the dark with respect to that.
Yeah, that's the other thing that all boils down up to now to transposon regulation somehow.
Absolutely.
Do you think you can also obtain similar effects if you have like pleiotropic changes in gene regulation instead of transposons?
Well, you could imagine. We know that there is epigenetic inheritance based on positive feedback or negative feedback loops in bacteria in particular, the lac operant, the lambda repressor that Geneviève alluded to. And there is no delimitation in these systems, by the way. The delimitation in bacteria does something else. It's there to help repair and recognize your foreign DNA compared to yourself. So yes, we could imagine that. But by and large, I think most of the kind of epigenetic phenomena that we are dealing with, which are based on delimitation, will be mediated by repeat sequences, and also in mammals as well. So if I'm allowed to say one thing, I believe that these things do happen in mammals. I've actually heard Azim Surani not so long ago saying that repeat sequences are not so much reprogrammed during the germline or in the early zygote development. In the early embryo development. So I believe that there you could also have accidental loss of methylation. But the mechanisms are much more robust than they are in plants in reestablishing methylation. So it will maybe last 2, 3, 4 generations. But it will very rapidly get back to the normal situation, I believe.
Eric Miska.
Yeah, I'm just wondering if you could comment on what is the status right now about making some of these epigenetic changes genetic. There's an old observation.
What do you mean by genetic? I told you that what I'm doing is genetic.
Change in DNA.
I'm provoking, sorry.
Yeah, no, that's fine. I mean that methylated cytosine has
so
a
epigenetic
different
variation versus DNA sequence-based variation.
Exactly. So in 1,000 generation of having a methylation pattern, you would have. Do you have enough time to mutate differentially between methylated?
Well, even if we can do 6 generations for Arabidopsis, I won't be here to comment in 1,000 generations. So we just have to speculate here. Yes, I believe that this kind of variation, epigenetically based initially, genetic in its properties, could be taken over by DNA sequence variation over a longer period of time. Absolutely. The difficulty is finding the evidence for that. And we know that DNA methylation has some properties with respect to mutability of the DNA sequence, but this is maybe not sufficient for us to capture such events as they occur. So we have no direct evidence now that something started as an epi mutation and then flipped into a DNA sequence-based mutation. Yeah. But I believe that this will be the case.
That was the last question. Thank you.